Make sure everything's correct. Okay, well, so 3 topics. It's easy and simple. PCA, t-SNE, and FATE. There are 3 different techniques for dimension reduction. And the PCA is oldest, and t-SNE may have 10 years history, and the FATE is only 3 years old. So 3 different algorithms have different ages, and they apply to similar situations. All the purpose is to do dimension reduction. So first, we want to know what it is and why we need that. I don't know how can I get rid of this bar here. Okay, it's good. So again, we come back to our single cell study context. We know we can get the UMI count matrix, which is gene expression data from many cells. Depends on how many cells you are targeted for per sample. Normally we have 6,000 or 7,000, even more cells. And for the expressed gene, we may have end up with 10,000 genes. So you have this big spreadsheet. From a data point of view, we will consider each column is a data point. In this case, is our sample or cells. And the rows are the variables or features to define the position of this data points, right? But conventionally, we also so once we have this, we are going to compare each of the cell and look at their relationship. If they are close to each other, we will put them together. And eventually we want to do the clustering analysis because we want to identify the different cell types. And so cells share the same expression profile are considered to be the same group of cells. So that will be the first step. We are going to reduce the dimension so we can somehow easy to manipulate the data. So you don't have to reduce dimension to 3 or 2 and you can see them. But normally it will help you to compute the distance, for example, if you reduce the dimension and get rid of some of the noise, because a lot of these features of gene expression doesn't tell you anything about the difference between cells. So we want to eliminate this. So another purpose of this dimension reduction is not only for visualization, but also try to get rid of this trivial information from the data to make this more informative. And then afterwards, you can so that's the pipeline. We will eventually reach out that stage. But of course, in the computer lab, you already, by clicking the buttons, you already go through all these steps. You can annotate the cell types, but internally, I want to show you how exactly this was done. And so you have UMI matrix. You want to get this reduced dimension representation of the cells data points. Then algorithm will help you to do this clustering. Remember, those clustering algorithms are unsupervised. So let's computer to decide whether they're similar enough to be grouped as the same group. Then afterwards, you can start to look at the marker genes. For example, this gene expressed highly in this green cell population, and this one maybe only highly expressed in this purple population. So eventually you will have some idea, okay, what those cell types are, and using your biology information knowledge, and you assign the annotation of cells. So today we are only going to do this step. So from a raw data matrix to do dimension reduction until you can see them, or you can easily to manipulate the data. Okay. This pipeline. So I'm going to transpose this because, well, in data science, we look at the column and like vertical vectors as the data points, but statistically you transpose it. So column becomes the feature or gene expression. So I use this transposed matrix to illustrate the high dimensional data, what that means. So if you look at only one gene, so now the column of the gene. So if you only look at this gene, TMEM 258, this transmembrane protein, I don't know exactly what it is. But if you only look at this one dimension, and then you will think cell number 3 and the cell number 5 are the same. Okay, so that's the resolution from one dimension. But if you consider 2 genes at the same time, and then you can project cell 1 to the coordinates like 8, 15, and cell 2, 5, 2, 1. Consider a 2-dimensional space. You will put these cells in that location based on this expression of 2 genes. So that gave you richer information because you consider multiple dimensions and characterize each cell. But now you can consider 3 genes, and then you still be able to plot them in a 3-dimensional space. Consider the first gene, you will put at X equals 8, Y equals 15, Z equals 11, and then somewhere in the space, and then you can point the second cell. However, once you go up to 3 dimensions, you only be able to plot them out. So at this end, you are dealing with high dimensional data. So you need to have this concept. See, we have dimension 0, 1, and 2, and 3, and we can see them. And 4, you have to imagine. But once you have thousands of genes, you have to consider a higher dimension of data. Okay. I hope everybody get this. And well, so our purpose is once you have been given this high dimensional data, a lot of cells defined by thousands of genes. So you get a cloud of data points. Are you going to be able to reduce the dimension to a certain level, 3 or 2, in order to visualize them? Or you don't have to go that deep, lower. In the middle somewhere, say, I want to use 20 dimension to summarize my data points. Then you have features and boil down from thousands of genes into summarized statistics, say 20 feature variables, and you can use 20 feature variables, and then your original matrix from thousands of cells, thousands of genes become thousands of cells, but 20 feature variables. So you reduce the dimension and make it much easier to manipulate. Okay. Questions so far? Okay, we are going to jump. Okay, so before that, that's dimension reduction. Just you have a 3-dimensional object, and you reduce the dimension by some kind of force, and then the data points are still there, but just visualize it differently. Depends on which angle and how you crash your data into a lower dimension. So that's why in single cell data, we use t-SNE a lot instead of PCA, because we realize t-SNE give you the better representation of original data points and fit our imagination better. However, before you do t-SNE, normally people will do the PCA first to initialize the position of the cell. So you do PCA first to put cell roughly by the similarity, and then you fine-tune the parameters, allow themselves to rearrange into the position that they looks like something like this. This certainly will be a cell from primary samples because they are different type and a lot of this heterogeneity in this data. Okay, so so PCA is the cornerstone of machine learning. As from the first lecture, I already emphasized this. I don't want to, I don't think I can overemphasize again and again. And if you go through today's lecture and you basically halfway through the whole machine learning field, I really believe so. And also, you already have all the piece information that you can put together to understand the PCA. So I'm going to first show you PCA today, and then we will mention t-SNE using the second half of the lecture. And then I will jump through UMAP because they are similar actually. And then we will mention the fit as I described in the outline today. So we will spend most of the time to study PCA, make sure you really understand. Please stop me in the middle once you have any questions, okay? So the first piece of the knowledge you need is this, okay? So you have your homework, you explain the projection of one vector to another. You can use dot product to compute it. Okay, I hope everybody understands this now. And think about this vector A and B. So the projection of A to B is the length of this here. If B is a unit vector means the norm of B equals 1. Okay, so if B is unit length, then you don't have to write this. So A and B, A product B is A's coordinate. So if you think B is x-axis, and then this position, the A will be, so that's x, A, y, A. This value is actually this one, right? And then you have y-axis. The y-axis, your projection on here is y coordinates. So that's the first piece of information we need to know. So dot product means you project your point based on that coordinate. This coordinate is defined by B and its orthogonal vector. B orthogonal vector. Okay. And then next is you need to know we can use squared matrix as a rotation matrix. Okay, you know that, right? So you can use this to multiply another vector to make the rotate. So rotate means the change the coordinates from original to the rotated one. So I'm going to illustrate this on the whiteboard, actually the blackboard here. Okay, make sure we know when you define this coordinate system using a squared matrix, you can change the coordinates of any data points from the point of view of new coordinates. So the whole point will be change the coordinates in order to display all the data points as separated as possible. Okay, now we are going to use blackboard to show the projection and the coordinate system, and also the change of the coordinate system. Okay. Any questions from an online student so far? Can I stop here? Okay. So, let's see. I have my current coordinate system like this. So this 1, 2, similarly. So I can define this coordinate system using 2 vectors. So my first coordinate system, I will use mu0 to define it. I will use 2 vectors. The first vector is 1, 0. Second one is 0, 1. Okay, 1, 0 is here. Okay, right? 0, 1 is here. So I'm using, by the way, I'm talking about this 2 dimensions. So I'm using these 2 vectors to define my coordinate systems. Now, if I have a point, say 1.5 and 2.5 here. I will call this my first vector, V1. (1.5), 2.5. Okay. So now I'm going to use my first vector to multiplicate. So if I use 1, 0, multiplicate my transpose this, multiplicate by 1, 5, 5, 2.5. So I will still get, oh, dot point. I will get 1.5. If you try this with dot point, this will be 1. So I'm doing is I'm project my midpoint to here. Not very good place. 1.5. Point. So my multiplication of this with my first vector is a projection of this. It's still 1.5. And this vector multiplication V1 will be the projection of this, and then of that. So I still have 1.5, 2.5, right? So that's my my point. So this unit vector does not give you any rotation.
Did you multiply the 2 matrices together? Just do
yeah, I
1,
can
0
I can
times
do
the
them
matrix.
together. So I'm going to do it together. So I can separate one, but I can do one step together, right? So I will. I think I have to do this mu0 multiplied by the transpose 1.5, 2.5. And after my rotation, I have to transpose to y. So there's some of this because it's not very easy to multiply. I think you have to transpose either this or that.
Oh, because when you did it, that's the 2 vectors together, you would just get one number.
Yeah.
And that would just get you either the x or the y.
So I can do this at the same time. So you project this to this vector and have this first value, and you project that to this vector and the second value. So if I do them together, I can using this to do this. Okay, now I'm going to try another vector. So this is boring because you multiply using this rotation matrix to calculated data point, you don't change anything, you still have the same value. So I'm going to use another one. So this one will be U1 as an example. Well, you have to take this as a so 1 over 2 minus. So this funny looking matrix is another example of my rotation matrix. But you can see, okay, this position is, you have it here, the circle. And this position is here. So I actually use another unit vector and also two of them combined to make a new coordinate system. So this new coordinate system is 45 degree turn from the original one. Okay, so now my new coordinate system is defined by this vector, these two vectors here. So now if I use my U1 to multiplicate by vector 1 transport, and you will get a new value, the value x. So this will become V1 will become coordinates have this, and this. The length of the first value will be from here to here. And the second value, that will be the ux. And your new y value will be, that's for V1. After you rotate and you have a new coordinate system, the length.
Wait, so are you not rotating the original vector U? You're just finding its current position and orientation in the new system?
That's right. So in green, what I'm writing here is based on the original. You have original. That's my original point of view. I have this point at 1.5 and 2.5. However, if I multiply this original point with my new coordinate system matrix, I will get the value which is from a viewpoint of my new coordinate system, and that's the length of here. So you update, the data point hasn't changed. You update your point of view, and your x, the coordinates of x v1 become from 1.5 from 1. So you change your point of view from your coordinate system based on the new location.
Right. But it doesn't change the actual vector itself. It just changes.
Change your point of view and say, okay, I use the new coordinate system to give a new coordinates values for that. So this new value you have seen here is after rotation, after you use new coordinate system, you get the value. Now you will see, okay, in original, and I have the distance from the origin to here is 1.5. And in the new coordinate system, my x-axis becomes longer or shorter?
Is it not the same that it's the x-axis?
It's actually longer. So this is this line here from that's x-axis. This value actually longer. It's actually 2.5.
Oh, you mean this position?
Yeah, the position. The position of the origin, the data after transformation actually from x-axis becomes longer. So this value, let me find out the value completely different. So the value actually becomes and this becomes that's my new coordinate system. Okay. So for this V1 data point, this original x-axis value is 1.5, but after I use rotated system, the x-axis value becomes 2.84. Actually, it's become longer through the x-axis. I don't know if I should try this in MATLAB or not. I think we are fine, right? Okay, somehow. Now, let's consider we have multiple data points. Now we have V1, we have V2 here somewhere. And also we have V3 here. Okay, if you have a 3 data point, and now I will ask, if we view this 3 data point from an original coordinated system by its x-axis, the level of separation from this to here, they are in the narrow range. However, if you view them from the new coordinated system defined by this vector, which is this right line coordinate system, they become most spread along the x-axis, right? So if you measure the difference between them, between V1 and V2 and V3, the difference between them become larger from point of view of x-axis in the new coordinated system. Is that correct? Yeah. Yeah. So the PCA is forgiving data points, say V. So in this case, we have 3, V1, V2, V3. If you have 3 data points for the given data, we try to find a new coordinate system defined by 2 vectors, V1 and 2. Because we are talking about the 2 dimension, because of 2 dimension. And 2, that's the dimension. One form is 2 by 2, 2 by 2. rotation matrix. So after our rotation, we will want this 3 data points to be separate as much as possible along one of the axes.
So you said U was a 2x2 matrix, right?
Yeah.
Okay.
So this is a rotation matrix. We want found. We will use the rotation matrix as a new coordinate system because it's particularly in a particular way to define and make sure this vector are orthogonal to each other. So you can use this combination. You can use this U matrix as a new coordinate system. So we want to found this new rotation matrix. To allow the new coordinates of the original data point become as much as possible separate, as large as possible. Separate.
Did I just consider one axis? So does it only want to separate them as far as possible on the x-axis, or does it also consider the y-axis, or
it will consider both?
Consider both?
Yeah, we consider first the left through x, then we consider second, because when you have high dimension, you have different ways to select the second axis, and then you have the third. So we will first consider the so-called the principal, the first component, which along the x-axis, then y-axis, then z.
If it does the z-axis, would it need to have another 3x3 matrix then?
Yeah.
Okay.
So now we are on this one, like all we are talking about the 2-dimensional system. So your rotation matrix or your new coordinate system are defined by 2x2 matrix. Well, if we upgrade to 3rd dimension, which I'm going to show you, then we need a rotation matrix 3x3. For our single cell study, if you have 6,000 genes, which means you have to define a unit matrix, rotation matrix, 6,000 by 6,000 rotate this.
And PC is just the algorithm to calculate or to maximize
to find this.
Okay. No.
So PCA is the algorithm to help you to try different ways to, I mean, not different ways. There are so many combinations to finding of parameters to define this new coordinate system to allow you to separate your data point as much as possible. Okay, so PCA is a way to find a new coordinate system for your data in order to show them more dispersed. Okay, to show the data point more dispersed. So now I'm going to use an example in MATLAB to show, okay, I can do a 3-dimensional PCA, okay? Good point. So I prepared a cloud of data. So on your screen, I don't know, do we have? Okay. Online student, are you okay? It's difficult, okay? So we have a data point, something like this. That's in the 3-dimension, okay? So I'm going to use PCA to find a unitary matrix. So I have 3 dimensions, and then my unitary matrix is this. So according to my PCA analysis, I need to rotate those data points. And based on this, so after rotation, I'm going to look at only the first and second column data. So I use my so my original data is saved in this vertex variable, and I use my U matrix to multiplicate it and to get my new coordinate system. So my S is my rotated data points. Now I'm going to plot them out only the first 2 dimensions, and you see that's a separation of the data. So that's 2 dimensions, and I project and rotate them, but I only look at the first 2 dimension data. I can still look at the 3-dimensional data. I can still do 3-dimensional. After my rotation. So it's still here, but the first and the second give you the most information. Okay, so the data points I get is from this T-port. So if you, I just added some noise, so we didn't recognize that the points are this T-port. It's actually the points on the surface of the T-port. So that's original. That's original projection. So X, Y, they. But if you do this PCA, again, I'm going to do the PCA for this and do the rotation and multiplicate original data and get a new data points and a figure. And that's my first and second components from that viewpoint, and that's a new coordinate system to give me this. So as I said, the PCA will help you to find the most new coordinate system, so you can display the most variability among your data points. So in this case, the most variable part is from this end to that end. Okay, globally, I mean, it's not consider only 1 or 2 points. Okay, on the white blackboard here, I'm talking about this 2-dimensional. So you consider rotate and use new coordinate system, and in order to look at x-axis. But here we consider x and y too, because the data is 3-dimensional. We want to reduce 1 dimension as an example.
On your U matrix in MATLAB, just to make sure, so you squared all those values and summed them, they would equal to 1, right?
Yes. So the U matrix is defined as a normalized vector matrix. So, and also the x and the y axis are orthogonal to each other. So you need to, yeah, there are some conditions on that. So they are not a random coordinate system. For if you do the 3-dimension, you have to make sure you have this x, y, you have to be the corner of the qubit. Make sure you cannot use a chain fit something like this to as your rotation matrix. You have to define something like that. Okay. Well, that's the way you see, I think I got this from the world market, and indeed, in this website, they only use one picture to show this T-port. So they select, not by random, they select this angle because you can display the variability among the data points on the surface of this. So it's consistent with what we discovered with PCA, right? Okay, our example is 2-dimension, but here is example from a 3-dimension. So you have a gene, you have cells expressed their position by 3 genes. So you have X, Y, Z, and then you want to find a rotation matrix which is 3 by 3. But after you rotate it, you view from the new coordinate system by looking at PC. So PC1 and 2 are the new coordinate system. And you also have a PC3, and they didn't show. But now you use PC1 and PC2 as a new coordinate system to look at your data. You see something like that. You separate cell. That's the most. Now, our question is, how are you going to find this rotation matrix? As I showed you in MATLAB, the code for PCA is so simple. So you just have your data points put in, and you want to get a U matrix. This matrix is a squared matrix, depends on the dimension of your data. If you have, so this dimension of the data in my example is n by 3, so 3 variable feature variables. So U will be 3 by 3. So that's all you want to get. So now the problem is, how are you going to compute it? Well, I have already showed you, you use a built-in function because many programs have languages also have this built-in function like MATLAB is PCA. I don't know in R, so all you need to get to compute unitary matrix and the rotation matrix. So I know how can I rotate my original data. You can also consider it as an optimization problem. So for this problem on the white blackboard, it's like I was given unitary matrix. I want to find a 2 by 2 matrix. So I have 4 free variables. Actually, they are not fully free. But basically, I want to find 4 numbers. So I can use them, put in my rotation matrix, so allow me to rotate my original data to show the largest variability among data points. I can have many different ways to select these 4 numbers. How are you going to do it? This is a kind of an optimization problem. You just say, okay, I want 4 free variables. Can you use algorithm to find it? The algorithm itself, the optimization problem, just search different combinations of all the numbers from 0.1, negative 0.1 combinations. It can help you to finally find it as long as you gave a so-called penalty function or objective function. So the objective function in our case will be the level of the variability of the points. So if you give me a matrix, I will compute the variability among data points and tell you how good your solution is. As long as you have this judgment, you have this objective function, you put that into an optimization engine. So there are algorithms to help you to do this. And then the most commonly used algorithm is to use eigenvector of the covariance matrix of your data to get the PC. So I will spend most of time soon to explain why this will be true. Okay, the eigenvector of the covariance matrix is the rotation matrix you want to find. So it certainly is a little bit of jump because the optimization probably is straightforward. As long as you know how to compute the variability after your projection of the new coordinate system, you can see, tell the optimization algorithm, can you just find the solution for me? So then you use black box machine learning and to get the data. But normally people don't do that because the next algorithm is more computationally efficient. You just compute the covariance matrix of your data, then take an eigenvector of those, and then you get the PC coordinates. Most of this lecture or online tutorial will end up here to tell you how to compute it, but nobody will go deeper to see why in this way you will get that solution. I will try to see whether I can get that point. And also PC, people use SVD, singular vector decomposition, which is another way to do it. Most SVD is so powerful. It's kind of the most universal powerful matrix decomposition algorithm. We will mention it later, okay, not today. There are also other ways, alternating minimization, and also there are other possible ways. Okay, so I've already showed you, and using example, if you use the built-in function in MATLAB or in other languages, you can just give this data matrix and get the coefficients. So the coefficient is n by n matrix, depending on how many features in your original X matrix input data. And these coordinates are the rotation matrix, okay, n by n rotation matrix. You can then compute using this rotation matrix to rotate the original data just by multiplicating the original data. You get the rotated coordinates. Well, there are other options if you want to go deeper, but that's for the optimization. So, for example, using this illustration, so we have original data that's two-dimensional are those blue points. And our new coordinate system is defined by this cross in the middle. So your principal components, the first x-axis you want to use, and the rotation is defined by this long rotated black line, and its orthogonal vector defined by this gray short line, okay? This cross in the middle. So we can select any of this along the rotation. So but our projection is to minimize the distance from all the data points to the new axis. So we want to minimize. Let me can stop. When we minimize the distance from a blue point to that, means we maximize the projection of our data points to the new one.
So is that what PCA is then, sort of? It maximizes the...
Yeah, PCA, try to see, this is a solution. At the end, we found, okay. At the end, we found, okay, this is the best PC along this.
So it has the shortest projection, but the highest variability along the curve.
Yeah.
Okay.
So when you have a short projection, the distance from the data point to that, the projection of data point actually is longer. So let me see if I can write anything here. So if you have, so that's our PC at the end we will found. So the data point, if one of the data points here, and its distance to, so you want to minimize this distance. When you minimize the distance, you actually maximize this projection. So that's the data point. So we want to minimize this to make this longer. Okay, so you do this for all the data points. So the collected projection becomes longer. So if you project every data point onto here, so the projection was this, so you make this longer. But when you have the major PC like this direction, then the projection on it is narrow globally. That's why at the end they actually found this longest. Okay, so that's a way to do the optimization. You just try them different ways of rotation. You can actually using the built-in function for optimization. This is found the minimum solution for the combinations of data. So we can try this. This is just you have you use built-in function get this rotation matrix, and then you use optimization way to get the similar results. Okay. So that's the optimization function. Okay, now we are going to move to the real solution people use is to search for eigenvectors for covariance matrix. Well, for this, you need to remember how to do this covariance matrix. And we quickly explained last time. And that's the original data. So you have 3 variables, 1, 2, 3. And in that case, your covariance matrix will be 3 by 3 at the end. So 3 by 3 at the end. Because you have 3 variables. So you centralize your data by making big A becomes here. So you centralize your data. In this case, they also scaled a little bit. So once you have done this, then your data matrix transpose and then time itself becomes the covariance matrix. So that's covariance matrix between variables, the relationship, correlation between variables, basically. In our case, you will compute a gene by gene coordinate matrix. So it's not a cell by cell anymore. We need that first. Okay, so express them mathematically. You have input data X. You have n samples, the key variables. In that case, the number of cells. That's number of genes. We remove the mean, we centralize the data. Then after that, this becomes centralized. Then you transpose this times itself and divided by n. That's covariance matrix. So covariance matrix become P times P. That's a gene by gene. And then once you have this coherence matrix, then you do the decomposition. You decompose them into this is called Eigen Decomposition. So you have your eigenvalue in the middle, and then that's the eigenvector matrix, eigenvectors. That's the PC. Okay. Another way to do it is to normalize your data, and then you try to decompose this covariance matrix, and then you largest eigenvector value will correspond to largest principal component with PC. Okay, so another way to show it, simply this way, this explanation is to consider the covariance matrix. In that example, we have a 2x2 because we are talking about this two-dimensional example there during data points, and then the covariance matrix is a 2x2 matrix. In this explanation, they use covariance matrix as rotation matrix. So you multiplicate your data point by this covariance matrix, you actually rotate your data points. The result of this rotation, and let me see the next page. And this method is explained later. The rotation of well, it's not a rotation in the matrix. So you use covariance matrix as a rotation matrix, but you rotate your newly defined coordinate system vector. So this is a little bit difficult to explain. So we use newly defined coordinate system as a rotation matrix to rotate my original data points. But in this solution here, they use covariance matrix as a rotation matrix to rotate the new vector you are going to use as coordinate system. And this rotation will help you to compute the overall variability in your data points after the rotation. Use your new vector system. I will illustrate it later. Okay. So basically here at the end, it will help you to know, okay, how are you going to compute this internally, and still not explain why you're doing this. Covariance matrix rotate your new axis system will help you to compute the total variability among your original data points. So I have to make the link after the break, okay? Again, just as trying to explain that link, I will illustrate on the blackboard. I will give you this as an example later, okay? Let's take a break and come back in 5 minutes. So we will come back at 6:11. Okay, I'm going to use this small example to illustrate that we can use two different ways to compute the variability of the data point regarding to the new selected coordinate system in different way. One is to use dot product of the projection, and others to use covariance matrix. And numerically, you will see they get the same answer at the end. Both of these two solutions give you the same variability level, 0.4138 at the end. So that's the first way to do it. The first way is here. Yep. That's the second way to use covariance matrix. Well, this data points, so that's my covariance way. I don't know covariance. Two different ways to compute the variability, total variability among data points after they project into new coordinated system. So the system I'm going to use is, so I just use this as an example, and that's my x, y. I have 3 data points. V1 somewhere here. Here. 2. With 3. So I have 3 data points on 2-dimensional space, and PowerPoint you will see that the real number in there, and also centralized. So this we have 3 points, and then I'm going to randomly select a new coordinate system. I only select x-axis. In this case, for example, I just try different ways to rotate my endpoint. So the most likely the PC should allow this way, right? But now I just select a random PC. I select this E as a new x-axis I'm trying. So you have y-axis somewhere here, right? So my original is 1, 0, 1. That's in the white, but now I select this E, and it's orthogonal vector as a new coordinate system. So now I'm going to compute the variability of 3 data points once they project onto my new coordinate system. So my first one will be, so V1 project. I have V1, I have a projection here. So that's my V1, right? My V2 is to do a orthogonal, and then that's my V2. V3 projection. V3. Right? If I use E as my new x-axis, my original data points, 3 of them project on myself, will have so V have 3 data points. V1, V2, V3. So I will do a dot product. Actually, I have to transpose this as E. Instead of doing multiplication, dot product, each one of them by E, I just use the whole matrix times E, and then I do a sum. So this is D1 plus D2, like this way. Actually, I'm going to square it. Okay, so you have learned this cosine and projection. So when I multiplicate each of my data points to E, I'm computing the projection of the data points onto this E, which is D1, D2, D3. Then I square them and sum. So I get this. So this is the total variance. Okay, so I use this left side and use this equation to compute the total variance of my data points projected onto my E, which is my randomly selected new coordinate system. So I can try different E. It doesn't matter. I can try different, and this my total variance will change once I change the E. Right? I can try a lot. At the end, I will find okay when my E point to that direction, I have large separation of the data. Yeah.
This is for the projection and not the distance, right?
Yeah, so that's a projection. So the projection on the distance. Yeah, the projection and the distance, they're orthogonal.
Right.
Yeah, so you have a larger distance and you have small projection. Okay, so on the right side, we will compute the covariance matrix. So the covariance matrix will be V times 3 transpose. That's divided by 3. So that's our covariance matrix. So in computer, I just showed you, and this equation just hold, and you can compute the covariance matrix. And this covariance matrix is a 2 by 2. A 2 by 2 matrix. Okay. For this particular example, we do have the covariance matrix. It's something like this, 1.7. So we do have this, our covariance matrix. This is a variable by a variable. So because you have two variables, so x, y, so that you have covariance matrix 2 by 2. But in that case, we can compute, if I use covariance matrix, that's called C, I'm going to use covariance matrix as my rotation matrix. For some reason, that's just the way it does. So I'm going to use my C. So I'm going to use C to rotate my E. My E is here. And use C as rotation matrix. Rotate it. I don't know now E become where. Maybe so the new E will point to point to maybe here. So this may be C times E. Then I'm going to do a dot product of E by that. So you have to transpose it in order to do this. So dot product of this vector dot product times this vector. Do you know what dot product means? Dot product is vector of E times CE cosine. Right? Dot product of these two will be the length of this times their cosine, the angle. But for whatever reason, the answer of this is total variance of your data. So at the end, this will be this, numerically the same. I don't know who figured out this trick, but eventually if you add this up, and you can do the projection of this, and also you can use covariance matrix to compute that, you get the same answer. Okay, so in order to optimize this, to find the E, which make the variance large, we can solve it by using C here. So make this large, to find an E, make this large. This is fixed because you have data, you have that. And then you found a way to maximize this equation will be the same as you maximize this equation. The red and green are the same.
Covariance is a measure of just the variability within the data, right?
Within the variables of the data, not the data itself. It's between the features define the data.
So between the X and the Y values.
Yeah. Yeah.
Are we multiplying it again by the E prime?
By E primer here?
Yeah.
This to compute. So you know this because this rotate your original this E. And your dot product with this, which means that is this. I don't know why exactly, but internally it's just compute.
I thought it was just C times E is the total variance. Is it the whole term together?
Yeah, this times 3 times together is the total variance. Because the dot product is a single number, so that's a single number. Right?
So it adds back another dimension to, because the dot product is like a scalar.
Yeah.
Okay.
So this is a vector. This is original vector rotated by your covariance matrix. And then you multiply it itself. Basically, it's like this. Okay, so we can confirm this. This use MATLAB. So we have normalized, centralized, and that's covariance using built-in function to compute and then validate. Okay, the covariance matrix we compute in this way. This is exactly the same. And the E is what we select, is this one here. We select, you can change this by any given E. So as long as it defines this coordinate, x-axis. Then you compute the projection, the sum of the projection onto it, and divided by 3 to normalize. And the numerically is the same as this. Okay, so that's why you can just focus on this part and forget about everything else. Then as long as you have covariance matrix, any new E you selected, you can use covariance matrix very easy to compute the total variance, total variability, the sum of all the points. Doesn't matter how many. Okay, so that's why in here you have this expression here. So you have a covariance matrix in the middle, then you newly selected coordinate system, which in the U matrix can be used to compute the total variability among data points. And then you try to maximize that. Well, I think the beauty of this is you can do this for millions of data points, and your covariance matrix may be very small, right? If you have millions of data, say, okay, you have 3 million of cells and define them using 50 genes, and your covariance matrix is only 50 by 50, so it's easy to compute. Instead of doing all the 3 million of data points to compute the total variability, you can use a covariance matrix to compute this total variability. Okay, let's move on. And so once you know you can do this, the Eigen decomposition will help you to find the largest eigenvalue, and their corresponding eigenvectors will be the coordinates you want found, because you know this relationship. Found this, give you the largest variability. And also, if you have a symmetric matrix, well, that's the case for our covariance matrix. It's always symmetric, and the eigenvector of the symmetric matrix is always orthogonal to each other. So that's mathematically guaranteed, and you will have the orthogonal vectors. So it's you don't have to worry, and you always get these systems orthogonal to each other if you do this in this way. However, the limitation of TCA is if you don't have this variability among the data. So like this, you have a hairball, and then your different ways to compute eigenvectors, eigenvalues are similar. So you don't have a good solution for this. It doesn't matter where you rotate it. So that's one limitation. Another is they're blended for this kind of a distribution. So you consider as a continuous distribution, but if you have data point somewhere here, some in a different patch, in this way, it will confuse the covariance matrix computation because they are not continuously distributed. You always have outlier points to drive your covariance matrix bias towards them. So in that case, it's not good enough. And also it will, because of doing this summary statistics, you ignore all the internal relationship between data points. For example, all these data points follow a sine curve, but PC will not understand it, only compute the covariance between variables. It will only found this line and gave you so there's no detailed description of the data points. So that's why even for the same data points, same data set, you get a TCA, something looks like this. And once you use other technology like TSNE, we are going to mention, it gives you the better resolution, especially when majority of cells look similar, okay? You can think about this as we do some kind of transformation instead of using the real distance. For example, if you use the real distance, the station between this underground will cluster together, not easy to visualize. But if you somehow transform them and make them more spread out, especially in the CBD area, and you have a better resolution. So we are doing something similar, not in principle, but effectively similar to that. So if you have a PCA, you try to get the data points more close to each other. And then once you use t-SNE to do that, you actually make this data point easy to spread out. The point is simple, and here, the first, we need to go back to 2003 for this paper, define the stochastic neighbor embedding algorithm. So the most important parameter here is the key. So if you have two data points in high dimensional, okay, I and j, whether these two are neighbors or not is defined by the distance between them and mapped to the Gaussian centralized distribution. Okay, and then for all, so for you look at point I and their distance to j, and then look at I to the distance of all other points. And then there's, you summarize this, and then the ratio between that is how likely you can, based on the distance between your I and j, you will consider j as your neighbor. So you have to consider not only the absolute difference, but also the other data points and relationship with it. And we can compute this P, which is the likelihood or probability of I and j are the neighbors in high dimensional space. And then you will do, so in this case, you compute the relationship between all the data points and their probability being neighbors in 3 dimensions. And then you compute projected 2 dimension and the lower dimension, another P, and here is called Q. And then you try to make this collection of distribution of the probability as much as possibly similar. So you want to minimize the divergence between them. If you minimize the divergence between them, that means if you found I and j are neighbors in high dimensional, they are similarly have a similar likelihood to be neighbors in the low dimension. So in this case, cat and kitten are the closer data points. So if I think cat are j, I and kitten are j, and in the lower dimension, cat and kitten are also close by to each other. They are more likely to be neighbors. So in that case, the probability of this P and Q matched. Okay, if you randomly put your cat and kitten away from each other, the likelihood between these 2 data points in lower dimension will be different from their original position in high dimension. In that case, you don't have a better small divergence. That means this embedding is not as good as non-random. Okay. So that's the idea. So you want to have a high dimensional space, the relationship between data points to be similar as low dimensional. So you have to compute the distance between data points there and convert that into probability. And then you optimize the location of your data points in low dimension to make their neighborhood relationship probability similar as in high dimension. Okay, that idea is not so hard, right? It just depends on how you compute the likelihood. I think then this algorithm was further developed and adopted the T distribution, and then by Van der Menten, and he published in 2008 and become a machine learning dimension reduction algorithm and popularly used in machine learning field. And later, so at that time, single cell data hasn't become available yet. So application of t-SNE was not designed for single cell data per se, it's for other high dimensional projected data, maybe from images, from semantic data and other applications. So this t-SNE is based on the stochastic neighborhood embedding algorithm, and they just select the T distribution to approximate the original distribution. There's a reason for that, and you mentioned very soon. So now, why is better than PCA? So when you have a complex object like this, so you would like to first detect its immediate local neighbor. That's the only way you can actually move with small steps to find your neighbors and move on in order to learn the whole structure of the complex shape. Otherwise, if you consider them together and all the data points in this non-linear distributed way, if you project onto lower dimension, they will superimpose onto each other, and you don't see the separation between them. You have to follow the trajectory of the data, and using, for example, this stochastic neighbor embedding to follow the link between data points in order to learn the structure. Otherwise, for example, if you directly project them linearly onto x-axis, and they superimpose onto each other, and of course, PCA try to separate them, but still, if you do some linear transformation like this, you do mapping, and you map this and into a separated location, and that gave you the better solution, right? So the T-SNE embedding just follow that way, and not straightforward to look at the neighbors, but look at the neighbors. So in this case, we have example in high dimension. You have an object in this red qubit, and then look at its neighbors and compute its distance from itself to all the neighbors, and then compute the total distance from itself to all the others. And it depends on the ratio. If you close by and you are most likely to be neighbors, so you compute this P. So in your left panel, this indicates, okay, we use this as a focal point and compute its distance to blue points, to red points, to orange points. And you see the distance to blue points are narrow. Okay, you translate into the high probability being neighbors because you use this Gaussian, this curve, right? The other way, if you are away from certain criteria and then away from this soft cutoff, then the probability being neighbors become much lower. Okay, for different groups, you can normalize it. So you somehow treat different group of data in the fair way. Okay, and then after you define this neighborhood probability, then you will map. So all the relationship between all the data points have been calculated. So you have a p-value matrix, and this matrix is pairwise between all the data points, right? So now you want to translate this into a low dimensional data point and also keep the same p-value matrix. And then you can pop up your data, and from the given P matrix and follow some kind of distribution. Let me show you, maybe I can, well, maybe not. So imagine you learn the the probability between point I and j from high dimension, and you see, okay, they are 85% to be neighbors, right? You keep this p, and you come to your low dimension. You first put your I in the low dimension, so you define I. Then you will see, okay, if I'm away from a 0.52 distance, then my j likely to be in that range. I will have 85% to be neighbor. So I will put my j in the low dimensional away from this I point 0.25 distance. Well, another point from the high dimension is only 0.30% probability to be neighbors. I will put in the low dimension far away from this, my data, based on this, okay, distribution and matched with their probability. The trick of this is when they learn the high dimensional p-value between data points, they use Gaussian distribution, they learn that. But once they want to reproduce into low dimension, they use t distribution. And the t distribution have this fighter likelihood to be in how to see. So it's less steep. So you see the blue curve here is Gaussian distribution. And once you're above distance above 2 in this case, then it's unlikely these 2 points to be neighbors. But according to t distribution, if you have a distance between 2 points are 2, then they still have 20% to be neighbors. So that's why if you found some slightly far away distance from a high dimensional, say 2.5 in high dimensional, you think these 2 cells will never be neighbors, but when you project using t-SNE onto 2-dimensional, you still put it closer by a little bit, okay, gave you 20% of higher chance to be neighbors. Okay. So because you reproduce the distance of your new point in the low dimensional space according to slightly different distribution of this shape. So at the end, you presented the distance between data points slightly different, but the overall relationship still kept, but the density just changed a little bit. Okay, so that's the key of the t-SNE. So you move in the low dimensional space, you move cells around until you reach some level of the global match between your probability of neighborhood, similar as your high dimensional probability, then you stop. Okay, so that's a t-SNE. So it involves this optimization procedure. So that's why you want to use PCA to first roughly place cells in by their neighborhood. So allow this optimization procedure easy to find the right position rather than purely random shuffle. The PCA is so quick, so it doesn't cost anything. So you have a initialized positions almost there. You just then optimize the position and fit the overall probability of neighborhood. Okay, so we are going to move to FATE, which is a new dimension reduction algorithm based on manifold learning. So in this case, it assumes your data is complex and also there are nonlinear relationship between data points. So something like this, okay? This shape here. This as an example. First, they compute the distance between all the points. Okay, you just compute pairwise distance, and then they apply a so-called kernel function. So the kernel function is a function will transform your distance into similarity. So so that's x-axis is distance. So if you have a very close by of these two cells and affinity or similarity is high, but after you're away from a certain distance, suddenly you have a drop of your affinity. So again, this is like a soft cut. If you're above this threshold, your affinity becomes really low. And in that case, the algorithm can trim off a lot of long distance relationship between cells. If they're far away to a certain level, I will cut and trim off this. At the end, you get this so-called, it's like a KNN network. Okay, so only cells will be neighbors linked if the distance is shorter than certain cutoff. And then from here, you start to follow the trajectory to move. So you can like a small agent to move from one cell to another, but I have to follow the path. The linked cell you can jump from one cell to another linked cell, but not cross its valley or, you see, Euclidean distance from one cell to another looks very close by, but once you have this builded network, you cannot jump from one cell here to a close by cell. You have to follow the trajectory, and that's the meaning of the manifold. Okay, you have this complex system, then these two cells may look very similar using Euclidean distance, but if you consider all the other population of cells, you have to go through different stage of cell from one cell become another. Okay, this happened actually in the cell differentiation. Okay, if you have a cell, in this case is a stem cell, and you have different trajectory to develop into different cell types, and in the middle, for example, here, you are ready to differentiate into skin cell and the liver cell. And then here you have something else, but they may similar, or not so different, but eventually they are quite different. You have to go through all the paths to the origin and then to come by. There's a way you can do regeneration, and maybe you can do the induced pluripotency. So you have iPSC, you can induce some skin cell back to stem cell and stem cell to divide into others. But that gave you the concept you have to go through, not just jump from one cell type to another, although they may look similar, right? So that's the idea about those manifold learning. And the paper is published in Nature Biotechnology. Here are some examples. They synthesize the data, and those data follow a distribution along this artificial tree. So this mimic manifold structure. So they know the colored data because they generate data, follow the tree, and then they know the true relationship between them. But if you use the PCA, you see, okay, you get this major PC XY, but your data points was crashed into, and you don't see this yellow and don't have structure at all. And the T-SNE gave you some of the separation because T-SNE really look at the neighbors, immediate neighbors, and try to, at the reduced dimension to reflect the high dimensional. So you still keep the global structure of data. And what they claim is the fate is much clearer separation, and they keep the trajectory of they designed for. Then they use real data. Here is embryo and differentiated data. So there are multiple data points, data types, and then reflect different levels of differentiation. And the PCA will mix up, still have some information in there. You have an early stage, later stage, and the T-SNE will help you to separate them into different clusters. But you see this relationship actually not kept as original differentiation steps, but the fate looks like the better reflects of the so-called ground truth because they know the marker genes and know this early development stage. And also data is collected from different days of embryos. Okay. So how this new algorithm is designed? As we already illustrate, we first compute a distance, pairwise distance between all the cells. Okay, we have data. We will transform the distance matrix into affinity matrix by using this kernel function. Okay, kernel function looks like a filter, and if you have a distance greater than a certain level, then your affinity becomes smaller. Otherwise, you have a high affinity, means these two are likely to be neighbors. So once you have that, they use a random walk to do a fusion process. Again, there are matrix multiplication steps to perform this. It's pretty easy, actually. And once you have learned this using diffusion process, basically they transform the affinity matrix again by using the neighborhood and the network structure. And then they get another so-called potential distance. And the last step will be MDS. Anyone still remember what is MDS? MDS stands for multidimensional scaling. So remember, we in the distance lecture talking about once you have a distance matrix between different train stations, then you can reconstruct the position of the station, different cities. For example, you know the distance between cities. So here they just use MDS. Once they have this potential distance, they just do MDS and get the projection of all the data points, all the cells in the map, basically. Okay, that's from their paper. So you have distance matrix, you build affinity, go through this kernel function, and then start to do random walk to build diffusion probability, and you get this distance, then you do MDS. Okay. Okay, that's all for today. And we talk about the PCA. I hope I gave you some idea, and your homework will be use your language to explain PCA. as much as possible to a certain level, and you understand and try to make your roommate maybe understand it, something like that. And then we mentioned the T-SNE, and very quickly go through fit. Okay, we will use them in single cell study. Okay, we use it every day actually. Questions? Well, good.